This topic we're going to see is pedigree analysis. So pedigree analysis is a tree, that is pedigree is a family tree drawn with standard genetic symbol showing the inheritance pathway for specific phenotypic characters. So here I had given a picture of a pedigree, that is family tree. So the two families and they are got their child, that is the daughter and son, for marriage. From that they are getting the grandchildren and great grandchildren. So this is a normal family tree. By this, the how the genes can be transmitted. So the forefather has a particular gene that was transmitted to the, that is, great grandson or daughter also. So that analysis is going to be done with the help of some genetic symbols to see the phenotypic characters. That is called pedigree. And pedigree analysis is the study of traits as they have appeared in a given family line for several past generations. So a particular trait is seen in one of the family tree to analyze the characters. So that to analyze the characters that has to be transmitted from generation to generation in a particular family. So that analysis is known as pedigree analysis. So you can see the symbols in this chart. So the normal square shaped box that is indicates male, and the circle indicates female, and this type is called mating type. That is, this symbol is mating symbol, and this is the symbol after mating you are getting the child's from. Okay, and this symbol shows the dizygotic symbol, that is a square and a circle shows dizygotic twins, and this one is monozygotic twins, like this means, and this is the sex, we can not expect unexpected sex, and this is the last one is the number of individual produced for a parents in their lifespan. Okay, and here this is a normal one, but this one is the affected one. So you can see the fully colored square and circle box, that is affected one, and this condition is heterozygous condition, but they are the carriers, and a dot in the circle shows the sex linked genes are the carriers, and a box with a line cross is death, and this symbol, that is a pin, that is, it looks like a bell pin, upside down bell pin like structure, shows the abortion. And this is Propelius type. This type is Propelius, and this is the cross. When we get one of the, that is, out of 3 children, one will get the Propelius condition, and this last one is consanguine marriage. Consanguine marriage is the relative marriage. Genetic disorders. So genetic disorder is a disease or syndrome. So it may be a disease which affect a particular family or a particular person through their genetic history, that is, through the generations of the generations it transmitted. So that is a disease or a syndrome. How it is caused? The abnormality in the DNA of an individual. So the DNA is affected. It causes genetic disorder. So abnormalities, how this abnormality is occurring? It may due to small mutation. Mutation means sudden change in the chromosome or a gene. So the mutation is seen in a gene. It may be addition, adding of a gene, adding of a, that is duplication of a same gene, or subtraction, that is deleting of a gene, or the entire chromosome will be deleted, or the entire chromosome will be added, or a complete set of chromosome is deleted or complete set of chromosome is added. These are the abnormalities in the DNA of an individual. So Then genetic disorders are of two types, Mendelian disorder and chromosomal disorder. Mendelian disorders. So Mendelian disorders, that is the alteration or mutation in a single gene, causes Mendelian disorders. So Mendelian means according to Mendel, the genes from the parent will be transferred to the offspring. So this is the Mendelian pattern. By this way, the disease, that is the genetic disease, is transmitted. Then it is called Mendelian disorder or Mendelian disease. So if a single gene is altered, that is altered or defected in the parent, it will transmit it to the child. So this is a Mendelian disorder. So some of the examples of Mendelian disorder is Thalassemia, Albinism, Phenylketonuria, sickle cell anemia, Huntington's chorea. So they may be dominant or recessive and autosomal or sex-linked disorders. So first we will see the Thalassemia. So this is a picture. I had given a normal Thalassemia, that is anemia. It can also known as anemia. So I had give the normal RBC structure, and this was the affected individual structure. Why it affects, that is in this normal you have 4 types of polypeptide chains. That 4 types may be alpha 2, that is alpha 2 and beta 2. So each 2 in normal one. Okay. But here either alpha chain or beta globulin is present. So here both are present in normal one. Okay. Both the alpha and beta are present in normal, that is hemoglobin. But in Thalassemia blood, either alpha or beta is present. So this leads to anemia. So what happens? Because either one of alpha or beta is present, the RBC is destroyed. So destroyed. So the RBC is destroyed. So due to the destruction of RBC, what happens? The anemic, that is the less count of RBC is there. This leads to anemia. And what is the reason for this anemia? The alpha has 2 genes, that is HBA1, that is HBA1 is hemoglobin alpha 1, and another gene HBA2, that is hemoglobin alpha 2. So these are the 2 genes responsible for alpha to produce the, that is alpha thalassemia. And in beta you have, that is the beta is responsible for HBBB. These are the only one gene which is responsible for not producing beta globulin. So in alpha stage the beta globulin is not produced, and beta stage the alpha will not produce. So this is the condition seen in the thalassemia. So due to this the anemia will seen, that is the alpha thalassemia. That is in alpha thalassemia the beta globulin won't be produced. So due to this what happened, that thalassemia is called alpha thalassemia, and it also known as Cooley's anemia. So you understand this thalassemia. So now I am giving this, that is it is an autosomal disorder. Autosomal means what? Means the body chromosome is affected. In body chromosome the recessive gene is seen. So due to this thalassemia occurs, and it occurs due to the gene mutation. So any change in a sudden change in the gene can also causes this thalassemia. So the gene mutation, there is excessive destruction, that is more destroying of RBC occurs. So due to this formation of abnormal hemoglobin is formed. Okay, so normal hemoglobin you have, as I said in the diagram, you have 4 that is polypeptide chain, 2 alpha and 2 beta chain. This is the normally present in the RBC. But in thalassemia, either alpha or beta will be seen. So they are in 4. Alpha means 4, beta means 4, 4. Only one type is seen in the thalassemia patient or person. Okay, so based on this hemoglobin chain, thalassemia is divided into alpha thalassemia and beta thalassemia. So the alpha thalassemia is controlled by 2 closely genes, that is linked genes, HBA1 and HBA2. That too where it is present in the human chromosome, the chromosome number 16 it is present. So the mutation or deletion of the 4 alpha, any one or all the 4 can undergoes to produce, that is alpha thalassemia in a person. And in beta thalassemia there is no beta globulin production. So the beta globulin production is affected in beta thalassemia, and it is governed by a single gene called HBB. Chromosome is where it is present. The HBB gene is present in the chromosome 11. So, and most common type of thalassemia is Cooley's anemia. And this disorder, the alpha chain production is increased. So, if the alpha chain, that is beta globulin, is affected and alpha chain production is increased, that leads to the RBC membrane also. The outer layer of the RBC is also affected. Next, phenylketonuria. So it is an inborn error. Okay, it affects the phenylalanine metabolism. So it cannot able to produce the phenylalanine metabolism, and it is an autosomal recessive gene. So what happens means, normal person, the phenylalanine, that is this phenylalanine is converted to tyrosine with the help of enzyme phenylalanine hydrolase. This is the normal procedure. But the phenylketonuria affected person doesn't have this enzyme, that is phenyl hydrolase enzyme. Phenylalanine hydrolase enzyme, and it is that particular gene is absent, that is PAH, that gene is absent in the phenylketonuria person. Normally the gene is present in the chromosome 12, that is seen for the hepatic enzyme phenyl hydrolase to function that is phenylalanine to, that is tyrosine. So the particular individual lacks this gene to produce the enzyme. What will happen? The phenylalanine won't convert to tyrosine. Tyrosine is an amino acid. So it converts to phenylpyruvic acid and its derivatives. So what happens? What are the characteristics if a person getting a phenylketonuria means? Severe mental retardation, that is the mental one, that is the nervous system will be affected. Mentally they won't be in a normal condition. High pigmentation of skin and hair, the coloring of a hair and skin may be dark in color. Pigmentation means color. Phenylpyruvic acid is excreted in the urine. So phenylketonuria, if they ask the question phenylketonuria, you have to write these points, that is it is an inborn error of phenylalanine metabolism. Autosomal recessive gene, that is the body chromosome, it is a pair of autosomal recessive gene. And the gene PAH, phenylalanine hydrolase gene, where it is located? It is located on the chromosome 12, and it is the hepatic enzyme. So if it is not present, in the chromosome 12 PAH, what happens means the phenylalanine won't convert to tyrosine. So, and the phenylalanine is converted to phenylpyruvic acid and its derivatives. So, due to this accumulation of phenylpyruvic acid, there are some of the symptoms is seen in the patient. Severe mental retardation is seen, high pigmentation of the skin and hair, phenylpyruvic acid is excreted in urine. Albinism. So, albinism, you can see here the picture. There is no any black color spot, that is the complete absence of melanin, even in hair, even in eyes also, that is the cornea won't be black in color. So where and all we get the black color patches in our compulsory, that part should be the black in color, that is melanin should be more in particular parts, that part also it is black in, that is it is white in color, there is no melanin pigment in the body, and this leads to the poor eyesight also. They have some problems, so we are going to see in detail. So, albinism is also an inborn error of metabolism, and it is also an autosomal recessive gene. So, melanin pigment is responsible for the skin color. Absence of melanin results in the condition of albinism. So, what happen means the 3,4-dihydroxy phenylalanine DOPA has to convert to melanin, and it is present in this 3,4-DOPA is present in the melanocytes. This product, that is DOPA, converts into melanin pigment with the help of enzyme tyrosine. So this tyrosine is seen in the melanocytes, and by seeing this chemical compound DOPA, automatically the tyrosine function on it and produce the melanin pigment. But this is not seen in the albinism person. So this conversion doesn't take place, that is the melanocytes fail, that is in melanocytes the tyrosine enzyme is absent and it failed to produce melanin pigments. So due to this, the melanin, that is due to the absence of melanin, albinism, there is no any part with the melanin in the body is seen, and they are very difficult. They have poor eyesight. Even the cornea is black. Think what will the iris is black. Iris is not in black color, white in color. What will, that is the poor vision. And the hair is, that is even in a younger stage, the hair is white in color, and they cannot able to see the sunlight properly. So the eyes will be affected due to the sunlight. Even the skin will be affected, and they are very poor. That is, their immune system is also affected in albinism people. So next, Huntington's chorea. So till that is whatever Thalassemia or that is Phenylketonuria, and the last one is albinism. These and all autosomal recessive disease. But Huntington chorea is autosomal dominant lethal gene in man. So they may ask in 1 mark. So Huntington's chorea is a dominant autosomal lethal gene in man, and it causes involuntary jerking of the body, that is the muscles, the jerking occurs not involuntarily, and there is a progressive degeneration, slow degeneration of nervous system occurs. So due to this, there is the mental and the physical deterioration of the person occurs, that is slowly. It's the first point itself, slow degeneration. So step by step the nervous system is affected. So due to this, the nervous system is responsible for the mental and the physical work. So it is affected. And the patient having this disease will die at the age of 35 and 40. Next, till now we see the Mendelian disorders. Now we are going to see the chromosomal abnormalities. So we all know the human chromosome is diploid in number. That chromosome number is 46. So the chromosomal disorders occurs, that is errors or anything occurs in the number or the structure of chromosome. So number or structure of chromosomes. And the error in the cell division also it occurs. That is, this abnormality can occur if the cell division, the error, that is for 23, 24 maybe occur, and less than 23 can also occur. That is an error in the chromosome. It occurs, how it occurs? During cell division it occurs. So, and the failures, that is the chromatids fail to segregate in meiosis condition, that is cell division, that may lead to one or more chromosome, that is called aneuploidy. Aneuploidy means the one or two chromosome fail to segregate, that is fail to separate during meiosis condition or mitosis condition. So that may lead to the adding of chromosome or deleting of one chromosome, that is called aneuploidy. And the groups of signs and symptoms together characterized, that is due to the particular chromosome, some of them have been grouped and their symptoms, signs have been identified, and they had having a particular syndrome, that abnormalities is called a particular syndrome. So in human beings, that we're going to see is Down syndrome, Turner syndrome, Klinefelter syndrome, Patau syndrome. So these are the examples for chromosomal abnormalities. First, we're going to see the autosomal aneuploidy in human beings. So there are so many autosomal aneuploidies. We're going to see in detail is Down syndrome and Patau syndrome. So Down syndrome is otherwise known as Trisomy 21 because it occurs in the 21th chromosome. You have the trisomic condition. Tri means 3 chromosomes are seen in the 21th chromosome. So what are the characteristics? Or the symptoms? So severe mental retardation, that is their mental condition won't be developed. It will be like a child condition or it will be a mental patient condition. So that will be seen. And defective development of central nervous system, CNS, think that the brain is not functioning. In the brain, the central nervous system only, we have the cerebellum, medulla oblongata, everything. What will happen? The entire brain is not function means the person cannot able to do the mental or the physical work. Increase the separation between the eyes. So our eyes have, in between our eyes there are nose. Okay, there is one nose. And in equal only they had divided. But this patient has that is vast. That is one eye will be somewhat bigger and the partition between the two eyes, that is the nose portion, is much larger and there is no equal placement of the eyes. And flattened nose, the nose is flattened. So because of the flattening of nose, the eyes are separated too much. Ears are malformed, that is malformed means having, not like us, it is having. Mouth is constantly open and tongue is protrudes. Protrudes means it is in a roll condition or it may be have any sprinkle like condition in the tongue, and mouth is always open. They cannot able to open, that is they cannot able to close the mouth. So this type of the symptoms are seen in Down syndrome. So they may ask in 5 mark about the characteristics of Down syndrome or trisomy 21. Patau syndrome or trisomy 13. So this is seen, this trisomic condition is seen in the chromosome 13. Trisomy means 3 chromosome in the 13th place. So it is due to the meiotic non-disjunction, that is meiotic translocation, that is enlarged male crossing. Due to that it is caused, and it is characterized by multiple and severe body malfunctioning as well as profound mental deficiency. So multiple problems may occur because it's going to affect the mental deficiency. That is, mentally we are absent. That is, mentally they don't know what they're going to do, what they are doing and going to do. So that condition is called, the entire thing is affected. Even the brain is affected, the mental position is affected. How they can able to live a normal social life? So it is affected. Next, small head with small eyes. The head is small. So there is no development of a brain. The brain development is very small. Then the head development is small and the eyes are small, and cleft palate. Cleft is seen in the face. And malfunctioning of the brain and internal organs. Next, second one is allosomal abnormalities in human beings. Till now you have seen the problem occurs in the body chromosomes, autosomal. Now you are going to see the problem occurs in the sex chromosome, that is allosomal. So during mitotic or meiotic condition, that is gamete formation, that sex chromosomes are affected through this disease or syndrome. Example, Klinefelter syndrome, Turner syndrome. First, Klinefelter's syndrome, double XY males. So addition of one X chromosome results in the karyotype we having only 46. But addition of X chromosome seen, that is for 46, one of the X chromosome is added, then we have 47, that is double XY condition, and this is seen in the males. So this syndrome contains 47 chromosome, that is 44AA plus XXY, additional one chromosome X is seen. So the symptoms they are characterized by sterile males. So this condition leads to the infertility of the male, no sperm production, and tall, they are over tall, they can be 7 feet, more than 7 feet also. Obese, they will be too fat, that is according to them normal, for normal weight, that is correct height and weight should be there, but in this person they have too, that is obese, they are too fat, and they have long limbs, high pitched voice, the voice is like an echo, like an it may be like a monster like voice. Underdeveloped genitalia. Genitalia is the sex organ, the sex portion, that is it is not well developed, and female breast is developed. So they are called as gynecomastia. That is gynecomastia. You doesn't that is the female breast development is, that is feeble. Female is not feeble, breast development is there. So the breast development is seen in male. So because of the presence of two X. So due to the 2X, they show some of the characteristics of the female. So like feeble development, but it is won't be the production of mammary glands like that. Turner syndrome, XO females, there adding of 1X in Klinefelter syndrome. But here 1X is 0. That is 1X is not there, absent. So loss of X chromosome. So that means how many chromosomes? 46 minus 1 is 45 X chromosomes. So this syndrome has 44 autosomes and 1 X chromosome, that is 44 AA plus XO. And they are also, as in the Klinefelter, sterile males. Here also they are sterile females. Symptoms is low stature, that physical nature, they look too short. Webbed neck, web. You know, web is seen in the frog's hind limbs for jumping, they are using like that. The neck is too conjuncted and it has webs. So webs in the neck. Web-like condition is seen in the web, and there is underdeveloped. The breast is undeveloped properly. And the gonads are rudiment. There is no formation of gonads. That is, rudiment means in the part, the part is not well developed. So, and due to this, gonads development not developed, the gonads is not developed, well, the menstrual cycle won't takes place. That is during puberty, that is during at maturity time, the puberty, the menstrual cycle doesn't take place. Thank you for watching the video. If you have any doubts, post your doubts in the comment box. I will clarify it. If you like the video, like, share and subscribe to Science Easy Tech channel.